Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Today we will start new video. If you like it, please like, subscribe and comment to receive more videos in the future. Let's start our video today. Here we go. Bismillahirrahmanirrahim. Walhamdulillahi wahda. والصلاة والسلام على من لا نبي بعده. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. In this lecture, we will talk about krebs cycle. This lecture is presented by Elham Omer, MSc Medical Biochemistry, Khartoum University. Let's see the objectives of this lecture first. By the end of this lecture, all the students should be able to define krebs cycle, identify the general properties of krebs cycle, recognize the general sources of acetyl-CoA, describe the steps of krebs cycle, calculate the energy produced per each turn of krebs cycle, and discuss the regulation of krebs cycle. This is Hans Krebs, the pioneer who discovered this cycle, and therefore it is known as krebs cycle. The first product of krebs cycle is citric acid or citrate. Therefore, it is also known as citric acid cycle. Sometimes krebs cycle is also referred as tricarboxylic acid cycle or TCA cycle, because the first product citrate contain 3 carboxyl group. احنا لازم نعرف 3 names لأنه الامتحان بيجي either about krebs cycle or tricarboxylic acid cycle TCA cycle or citric acid cycle. Definition: it defined as series of enzymatic reactions in the mitochondrial matrix which oxidize acetyl residue of acetyl-CoA. The acetyl-CoA consists of 2 parts, acetyl and CoA. It oxidizes acetyl residue into 2 molecules of carbon dioxide, this first molecule and this is the second molecule, and releases reducing equivalents in the form of NADH, nicotinamide adenine dinucleotide, and FADH2, flavine adenine dinucleotide, and energy in the form of ATP or GTP. The differences between ATP and GTP: ATP adenosine triphosphate and GTP guanosine triphosphate. Both is the currency of energy. And regenerate oxaloacetate. The oxaloacetate which enters the cycle in the first reaction is regenerated in the last step. That is why it is known as cycle. When we talk about glycolysis, glycolysis is the pathway because it starts with glucose and end by pyruvate, different product. But this cycle start with oxaloacetate, and the oxaloacetate is regenerated again in the last step. General properties. Number 1, it is the mitochondrial pathway. This is the structure of mitochondria. It consists of 3 membranes: outer mitochondrial membrane, inner mitochondrial membrane, and the matrix. The enzymes of krebs cycle located in the matrix, either free or attached to the inner mitochondrial membrane. Number 2, it is the final common pathway for oxidation of carbohydrate, lipid, and protein, because monosaccharide like glucose and fatty acid and amino acid are metabolized into acetyl-CoA, which enter krebs cycle for complete oxidation into carbon dioxide and water. Number 3, it is amphibolic pathway. It occurs at the crossroad of metabolism linked between anabolic and catabolic pathways. Some pathways originate from Krebs cycle and some pathways end at the Krebs cycle. It has a central role in gluconeogenesis. The synthesis of glucose starts from Krebs cycle. Also, it has a role in lipogenesis, the synthesis of fatty acid. Citrate from Krebs cycle go outside to the cytosol and cleaved by citrate lyase into acetyl-CoA and oxaloacetate. Acetyl-CoA is the building blocks for fatty acid synthesis and cholesterol synthesis. Also, it has a role in interconversion of amino acid. It has a role in transamination and deamination. For example, alpha-ketoglutarate from Krebs cycle, when it undergoes transamination, it converted into glutamate. That means the alpha-ketoglutarate, when it accepts amino group, it converted into glutamate. And glutamate, when it lost its amino group, is converted back into alpha-ketoglutarate. Reactions of the TCA cycle. It is a series of 8 reactions in which each molecule of acetyl-CoA is oxidized into 2 molecules of carbon dioxide, 3 molecules of NADH, 1 FADH2, and 1 GTP. General sources of acetyl-CoA. Number 1, pyruvate oxidation, and this is the main source. As we said, in glycolysis, pyruvate under aerobic condition will be converted into acetyl-CoA by pyruvate dehydrogenase complex. Fatty acid oxidation. During the oxidation of fatty acid, there is the production of acetyl-CoA. Ketone bodies oxidation, individual amino acid oxidation, and those known as ketogenic amino acid, and ethanol oxidation. Reactions of Krebs cycle. As we said, it consists of 8 reactions. In the initial reaction between acetyl-CoA, which contain 2 carbon atoms, with oxaloacetate, which contain 4 carbon atoms, form citrate, which contains 6 carbon atoms. This reaction is catalyzed by enzyme known as citrate synthase. In reaction 2, the citrate, which contains 6 carbon atoms, is isomerized into isocitrate by enzyme known as aconitase or aconitate hydratase. This enzyme removes the water from citrate. It removes hydroxyl group from carbon 2 and hydrogen from carbon 3, and then adding water back at isocitrate at different position. Therefore, isocitrate is the isomer of citrate. In reaction 3, the isocitrate, which contains 6 carbon atoms, undergoes dehydrogenation, removal of 2 hydrogen atoms from carbon 3. This reaction catalyzed by isocitrate dehydrogenase. The 2 hydrogen is taken by NAD plus to produce NADH, and this through the respiratory chain produce 2.5 ATP. In the first reaction of dehydrogenation, oxalosuccinate, which contains 6 carbon atom, is formed and remain enzyme bound. Then it undergoes decarboxylation, removal of 1 carbon atom in the form of carbon dioxide to form alpha ketoglutarate, which contain 5 carbon atoms. In reaction 4, the alpha ketoglutarate, which contain 5 carbon atoms, It undergoes oxidative decarboxylation in a reaction catalyzed by a multi-enzyme complex known as alpha-ketoglutarate dehydrogenase. This enzyme contains CoA SH as cofactor. In oxidation, the oxidation means removal of hydrogen. This hydrogen from CoA-SH is taken by NAD plus to form NADH. This is the oxidation. Then also the alpha-ketoglutarate undergoes decarboxylation, removal of one carbon atom in the form of carbon dioxide to form succinyl-CoA, which contain 4 carbon atoms. In reaction 5, succinyl-CoA is converted into succinate by enzyme known as succinate thiokinase. It is also known as succinyl-CoA synthetase. Succinyl-CoA has a thioester bond with very negative free energy of hydrolysis. The bond found between carbon 3 and the sulfur of CoA. This energy of succinyl-CoA thioester bond is used to generate GTP from GDP and inorganic phosphate. This is the only example in the citric acid cycle of substrate level phosphorylation. In reaction 6, the succinate undergoes dehydrogenation forming fumarate. Is catalyzed by succinate dehydrogenase. Dehydrogenation means removal of hydrogen. The 2 hydrogen from carbon 2 and carbon 3 is taken by FAD, the coenzyme of succinate dehydrogenase, to form FADH2. And this through the respiratory chain produce only 1.5 ATP. This is the only citric acid cycle enzyme that is tightly bound to the inner mitochondrial membrane and considered as complex 2. In reaction 7, fumarase or fumarate hydratase catalyzes the addition of water across the double bond of fumarate. Here in fumarate there is a double bond between carbon 2 and carbon 3. This enzyme add water to break the double bond and form malate which contain hydroxyl group at position 2 and hydrogen at position 3. In the last step, malate is again oxidized by malate dehydrogenase into oxaloacetate. The oxidation here occur at position 2. 2 hydrogen from carbon 2 is taken by NAD plus to produce NADH plus H plus. By this step or by this reaction, the cycle is complete and can start again from the beginning. Metabolic energy of Krebs cycle. The metabolic energy is produced at 2 levels: substrate level phosphorylation and oxidative phosphorylation. We mean by substrate level phosphorylation the production of ATP directly through the reaction, and the oxidative phosphorylation production of ATP through the respiratory chain. This figure shows the structure of ATP. As we know, ATP are referred to adenosine triphosphate. It consists of adenine base and ribose sugar. Both form adenosine. When this adenosine combine with one phosphate group, It's known as adenosine monophosphate. When it combines with two phosphate group, it's known as adenosine diphosphate. When it combines with three phosphate group, it's known as adenosine triphosphate, and this is the currency of energy. ATP production. 10 molecules are formed per turn of the TCA cycle, because each turn produces 3 molecules of NADH, 1 molecule FADH2, and 1 GTP. Each of 1 NADH through the respiratory chain produce 2.5 ATP. 3 multiplied by 2.5 equals 7.5. And the FADH2 produce 1.5, the total 9, and 1 GTP, the total is 10 molecules of ATP. This figure shows the complete oxidation of glucose by glycolysis and Krebs cycle. As we said, in glycolysis under aerobic condition, it produces 7 ATP. Why? Because it produces 2 molecules of NADH. Each of them through the respiratory chain produce 2.5 ATP. The total is 5. This it depends upon whether it enters the respiratory chain through the malate shuttle. But when it enters the mitochondria through the glycerophosphate shuttle, it produces only 1.5 ATP. The total is 5. So the total of ATP produced by glycolysis under aerobic condition is 7 or 5, depending upon whether this NADH enters the mitochondria through the glycerophosphate shuttle or malate shuttle. Also, there is the 2 molecules of NADH comes from the reaction which converts pyruvate into acetyl-CoA. 2 molecules through the respiratory chain produce 5 ATP, and each turn of Krebs cycle produce 10 molecules of ATP, and we have 2 molecules of acetyl-CoA, therefore we have 20 ATP. So the total is 20 plus 5, which comes from reaction which convert pyruvate into acetyl-CoA. This is 25, and plus 5 or 7, it depends. If it enters the respiratory chain through the malate shuttle, therefore we have 7 ATP. The total is 32. If it enters the respiratory chain through the glycerophosphate shuttle, the total is 30, because we have 20 comes from Krebs cycle, and 5 which comes from pyruvate into acetyl-CoA, this reaction, and 5 which comes from glycolysis. Regulation of Krebs cycle. The reaction which convert pyruvate into acetyl-CoA is catalyzed by pyruvate dehydrogenase complex. This enzyme is inhibited by ATP, acetyl-CoA, and NADH. When we see circle with negative, this means inhibition. Circle with positive, this means activation. The ATP, the aim of this reaction is production of energy. When there is enough energy, this will cause feedback inhibition of this enzyme. Also acetyl-CoA, because it is the end product, because this enzyme convert pyruvate into acetyl-CoA. Enzyme which convert isocitrate into alpha-ketoglutarate is known as isocitrate dehydrogenase. This is inhibited by ATP and NADH and activated by ADP. The reaction which convert alpha ketoglutarate into succinyl CoA is known as alpha ketoglutarate dehydrogenase complex. This is inhibited by ATP, succinyl CoA, and NADH. These reactions in Krebs cycle is inhibited by ATP because the main objective of Krebs cycle is to produce ATP. References for this lecture include Harper's Illustrated Biochemistry, Stryer Biochemistry, Lippincott Illustrated Reviews Biochemistry, Yosemite Kaplan. عندنا more references عندنا لينينجر ديفلين لو عايزين more details. النت كمان مفيد في بعض الأشياء عندنا على قناة اليوتيوب في simplified videos ممكن تكون مفيدة. تم الكلام وربنا محمود وله المحامد والعلا والجود ثم الصلاة على النبي محمد ما ناح قمري وأورق عود. This is all I can say in this lecture. I hope you understood. Thank you for listening and you have a great day.